back to the session. Our first speaker is Shobhona Sharma. She joined the molecular biology unit of TIFR as a PhD student in 1975 and earned her degree in 1981. She continued here for a brief postdoctoral fellowship and then went on to New York University Medical Center and finally to Duke University Medical Center. She came back to the molecular biology unit as a faculty in 1987. In this time, she transited from physical chemistry to molecular biology of plants to malaria parasite molecular biology and immunology. After coming here, she has set up a lab on the study of malaria parasite and some other protozoan systems. She has published extensively on parasite biology and has also filed for patents. She's a recognized expert in the field of parasitic molecular biology and immunology and has contributed as a member of several scientific advisory committees and governing bodies. She has mentored and supervised the research work of nearly 30 PhD and MSc students who have carried out the legacy of being successful scientists all over the world. She has also been involved in women in science activities and indeed figures in the famous book on women scientists from India, Lilavati's Daughters. Shobhona Sharma.
Thanks very much for this kind introduction. As you see over here, I'm going to talk mainly about the pioneering of the molecular biology, and the organization is over here. So history will form the major part of my talk, and then of course I'll talk about research activities in parallel and some of the teaching and workshops, and I'll talk about the current status at the end, and obviously that will be quite incomplete as you'll see. Okay, so history, so biology is at 60 years, not 75. Obaid Siddiqui joined in 1962, and the story is very well known. He was handpicked by Bhabha. Most people in those days were handpicked by Bhabha. Subsequent members of molecular biology unit were handpicked by Obaid. The group was then called Molecular Biology Unit, and I will comment on that also in subsequently. So Obaid started research in Tata Institute. Of course, many physicists who were asked by Bhabha actually said that this is not the right stage for any biology person to join. Where are the resources? What will he do, etc., etc. But nevertheless, Homi Bhabha had decided based on various criteria. So he had joined, and he had narrated these stories that initially he had only, he didn't have an autoclave where you could sterilize things. So they used pressure cookers and so on. So all kinds of jugaad, which TIFR is very famous for, he used, and he started with bacterial and fungal genetics and did seminal work in the first 10, 12 years. In that field, Ram Naresh Singh from BRC training school also joined. But the trend for molecular biology unit in early days were individualistic. Everyone did her own work, and you'll see that. So such a subject, molecular biology, did not exist in any biology syllabus. Even now, you don't find it as a subject per se. These traditional biology subjects, botany, zoology, microbiology, were getting modernized in 60s, and some places were calling it life sciences. But Obaid had this vision of biology at the molecular level, so he called it molecular biology. And he also convinced University of Mumbai to start a molecular biology department. We all earned our degrees in molecular biology. Nowhere else in the world do you earn your degrees in molecular biology. Now, deemed university gives it in biology, so I'll come to that. So soon after Obaid, it was Dr. P.K. Maitra who joined along with RN Singh. RN Singh joined actually earlier, P.K. Maitra joined later. And he worked on utilization of glucose by yeast using genetics and biochemical tools. His personality was such that he would be in his lab all day. We had a common lab and he could see us from his office, not office, from his lab bench all the time. And we were extremely upset that we couldn't do things which otherwise, at night, we might want to do, sing songs or whatever, have the radio blast on and so on. He was always there in the lab. Subsequently, and of course, they did fantastic work on glycolysis. I'll come to that. UN Singh, M.R. Das, Kenkare, Johri, and Babu joined. And initially, it was these scientists who played formative roles. Anil Sadgopal also joined, but he's from Caltech, but he soon left. He had completely leftist tendency and wanted to, teach science to rural school students as well as farmers over there through innovative methods. And this was Kishore Bharati. Eklavya is the fallout of that which exists even now. I don't have a picture of Anil Sadgopal. I'm sure we can get it. He was associated with Central Institute of Education in Delhi. Umakant Kenkare and M.R. Das. So he used enzymes and studied their structure-function relationships and regulation. M.R. Das worked on RNA tumor viruses, which was a very hot topic at that point. He worked with Saul Spiegelman and came back and continued to collaborate. And there was a lot of clamor among students to work with M.R. Das at that time. But in early 80s, he left TIFR. So Obaid's dominating, personality was very difficult for people who wanted to, start domains themselves. So many people have left, and M.R. Das also left and joined CCMB Hyderabad and found his own institute, Rajiv Gandhi Center for Biotechnology in Trivandrum. But TIFR is also, this was one of Homi Bhabha's expectation that TIFR will actually have scientists who will move out in the rest of India and start important institutions, directions of work. Manmohan Johri, his picture is put up outside. He is as one of the unknowns, but he did very, very good work over here. He came with plant molecular biology, and Obaid was not at that point looking for plant molecular biology. It's a difficult field to start with because growing plants, you need space, resources, etc. But he met Obaid and Maurice Fox, both of them at MIT, and had a day-long interaction, and then Obaid offered him a job. And after joining, he tried many projects, which were, some of which did not quite work out, but finally he settled with the project of moss cell differentiation, which was essentially done very nicely because normally moss grows on solid agar. That's how they're grown in the lab. And any study is very difficult. You can do microscopy, but it's very limited. But he developed the technique of growing liquid cultures of moss cells, and we could follow that for the cell differentiation and development, and that gave very unique features. I'll come to the work actually. Babu. So these were the early people. So he was a PhD student. He also came from BRC training school, did the PhD in theory physics in TIFR, but he was attracted by the possibility of genetics and molecular biology. And after a postdoctoral stint, joined MBU and brought with him the system of nematodes, C. elegans, and worked subsequently with fruit flies as well. And he was involved in neurobiology, developmental biology, mainly using the tools of genetics. I have never seen Babu do anything but genetics, and he would always leave dirty biochemistry to other people or collaborators or students. He always worked with genetics. In the late 80s, again, Babu decided to I mean, you know, he was a very restless personality. So he felt that he was doing the same thing over and over. And that why is it that we could not buy reagents for recombinant DNA technology, which was coming up at that time? We had to import all of it. So he took this giant leap of moving to Bangalore and starting this company, Bangalore Genie. And all of India is very indebted to him for this company, because now they could buy it in Indian rupees, the reagents, and it was really one of the first kind for India. Subsequently, 72 to 82, Krishnan and Vidyanand Nanjundaya joined. Krishnan joined in 77, Vidyanand joined in 1980. And both came with physics background. You can see all of, other than Manmohan Johri, P.K. Maitra, Kinkare, they all had chemistry background, or like Babu, Krishnan and Nanjundaya had physics background. So not many traditional biologists joined at that time. Even among students, that used to be the flavor. Most of us came from chemistry, physics backgrounds. So Krishnan started with snail biochemistry and neurobiology. And many of you might be familiar, because soon they escaped the animal house and was all over the colony and, you know, moving around very freely. But subsequently he moved to Drosophila, and Vidyanand studied cellular slime mold. This is an amoebic organism that lives as single cells when food is plenty. But then the moment starvation sets in, they actually aggregate to form one body, and this body moves as a slug and develops apical and polar specificities. One part becomes fruiting body, the other part remains the anchor. And so it is a very attractive system for looking at signals that would cause these differentiations. So 72 to 82 also heralded the neurobiology in MBU. So Obaid went for a sabbatical study and came back with the idea that there should be higher organism. Bacteria you cannot is limited only to very basic cell biology or molecular biology. So he started fruit fly neurobiology, and Veronica, who was one of the PhD students who joined in 1976, pioneered the studies. And of course, famous are the home-built, glass-blowing section-built Y-maze apparatus, using which they got wonderful, you know, so you feed in the flies and you put air on one side and the particular chemical on the other side, and see whether flies move to this end or that end. And you count the numbers and you can see whether, so you know what the normals do, and you can generate mutations and look at what the mutants may do. And she joined as a faculty in 1982. A whole bunch of our students joined the department at that time. I have already talked about Veronica. LC Patty joined in 76. Vijay joined in 83 or so, and I joined in 87. Zita Lobo moved from scientific to academic stream. Here is Zita Lobo. And Gautam Jarori moved from solid state physics. So he was a member of solid state physics. I noticed that his name never came up. Kasturi's came up, but he was a solid state physicist until about mid-90s. And then he moved to MBU. So Gautam in the early days. You know. So the only non-TIFR alumni inducted during this period was Subhrata Tripathi. 82 to 2002, molecular biology flourished in TIFR Mumbai, but space constraints were very real. And Ubaid wanted to expand. There were many people who could join and join the existing programs as well as other programs, lines of work. So he realized that he could not expand in here. And in Ubaid's own words, the idea that TIFR should start a center for biological research was mooted in 1982, following a suggestion by Ramaseshan. But that could not happen, that is location at IISc campus. But the Planning Commission in 1984 agreed to fund a center for fundamental research in biological sciences. And the center was to function as an autonomous unit under the aegis of TIFR and conduct fundamental research and training in areas of biology at the frontiers of knowledge. So I believe there were questions about why biology should expand, but clearly Ubaid could convince them. And after several years of research, University of Agricultural Sciences at Bangalore offered an attractive site on their campus. And this was a very good move because it wasn't new land. New land had a lot of problems. So it was within the campus, their existing campus, and an MOU was drawn up and signed in February 1991. Since the funding had already started, Ubaid had already started recruiting for NCBS. And Jayant Utkankar, M.K. Matthew, Panikar, these were some of the early appointees who worked with members at MBU in the department, in the labs. There were lab spaces assigned to them. And only in 91, 92, they moved physically. So first to the IISc building, which was available to them, and then to their new building. And of course, from the department, I forgot, Geti Hassan also joined around 1987, 88. She was a postdoc with LC Padhi, and she became a permanent member. And these people moved from MBU to Bangalore. Meanwhile, in this period, B.J. Rao, Rohit Mittal, Shubha Tole, and Vidita Vaidya were inducted in TIFR Mumbai. This wasn't easy. Once Ubaid decided a new center would be formed, all the good cases, quotes, he would take the files and he would keep them with him for NCBS and would not allow molecular biology unit to appoint anyone. Dr. P.K. Maitra, who was an excellent scientist, but did not have the kind of administrative acumen to override this or to search for people on their own. And of course, who could match Ubaid in the charisma and attractive power? So we had a tough time. We also had a tough time because it was expected that biology would die over here and everyone would move to Bangalore. And why recruit people? But thanks to Porter Commission, I think, I'll come to that. So nevertheless, we could induct excellent people over here who continue to doing work over here and excellent work at that. So NCBS, coming back to the history. So this is from his own write-up, Ubaid's own write-up. He says that he went by the guidance of Flexner, who proposed the creation of Princeton, the Institute of Advanced Study in Princeton, and that it should be small. Its staff and students or scholars should be few. The members of the teaching staff, while freed from the waste of time involved in administrative work, should freely participate in decisions involving the quality and direction of activities. The living conditions should represent a marked improvement over contemporary academic conditions, and its subject should be fundamental in character, and it should develop gradually. And that was something he adhered to, because initially there were about handful, 8 to 10 scientists, and it slowly grew. And that has its own advantages, because he could really look at certain fields and induct. So of course, this is the current status. He was the center director until 1997, after which Vijay, and then now it is Jitu Mayor. Hyderabad, I have just one slide because it's going to be talked about separately. And the point is that, and this is again from the director's note for the Hyderabad. And I just want to point out that currently there are about 30 faculty members, of whom about 50% are engaged in some form of biology research. Some of them are totally molecular biologists or immunologists. So Pratim Majumdar, who was a student of Shiv Prasad from NCBS, is one of the first biologists. And now there are Monica Vij, who's an immunologist, and Adish Dani. So some who are basically molecular biologists, but then there are many others who have, who collaborate. So they are from physics and chemistry, and there's a very viable, there are very viable collaborations. So research areas and activities. Do tell me when I have 5 minutes left, okay? So Ubaid encouraged all faculty members to find their own questions and their own models. And this was the beauty of TIFR as a whole, I think. Basically, people were free to do their own work, whatever kind of science they wanted to do. So the early areas, 62 to 82, were bacterial fungal genetics and biochemistry. So Ubaid worked on Aspergillus, enzyme kinetics by Kinkare, viral molecular biology, plant development, and neurobiology. And he tried to unify these into neurobiology and molecular biology. This was because the planning commission didn't want to hear about 10 different projects. So he realized that some kind of grouping would be useful. So neurobiology is very clear. The rest of the people got clubbed into molecular biology. So molecular biology has been a topic that was very useful. He was also keen on group of scientists working with infectious diseases immunology, and that's how myself, Sudhir Krishna, and Apurva Sareen were recruited. And subsequently, Sudhir and Apurva were clubbed into cell biology. I'll come to NCBS. They were in NCBS. In the 90s, the term molecular biology became highly questionable. It got associated with recombinant DNA biology, which is genetic engineering, and it lost the general value. In 1997, somebody was asking when the structure happened. So 1997, the Porter Committee came and gave their report. There was a large-scale review. And instead of groups and units, so the only school that existed at that time was School of Mathematics. The rest was called School of Physics or Physics Faculty, which was, of course, not so suitable for us and for many others. So then 3 schools were formed: computer science, mathematics, and natural sciences. And the groups and units got designated as departments. So molecular biology unit became Department of Biological Sciences. And at that time, I remember we were quite unhappy about this change, and many of us would call it a dubious department and so on. So in 2002, TIFR became a deemed university, and under the biology subject board, the degree which is conferred now is biology. So the name does matter. I mean, even though great poets have said what's in a name, but name does matter. Okay, I am now showing some slides prepared by my colleagues when we had to prepare for NAC. And so I just thought these were beautiful, and they were made in 2016. So NAC is the Committee for University Accreditation. So at that time, we had all these model organisms, and I just want to, and we grouped, as you see, it was nice to group. The same person would occur in many groups, depending on what their work is. But I want to talk about the model organisms. So fruit flies system was brought in by Ubaid in 70s. Yeast was worked by P.K. Maitra, but when Maitra and Zita Lobo were no more, then this sort of died, and then it was revived by Himanshu, and then again he left, so no more yeast work. These are red cells containing plasmodium. These are zebrafish. Zebrafish also, UN Singh, Uday Narayan Singh, decided to work with zebrafish, and for about 6 months we had this beautiful aquarium in the common lab, and he did some developmental biology work but later gave up. Anyway, he moved out to Delhi University. But now, I mean, subsequently it was brought back by Mahendra Sonavane, and Shailaja also worked on it. Mouse system, of course, thrives the most because many of us worked using the mouse model. Shubha and Vidita are the prime users. Ullas, myself, we were also extensive users. This is the cellular slime mold. So Vidyanand brought it with him, and he left in late 80s to join IISc. So this work stopped, but then Roop Malik came in and did a good bit of work in using cellular slime mold. And so it came back. So model systems keep coming and going. C. elegans nematodes were brought by Babu, and it was given up early, but then Sandhya Kaushika brought it back, and a good bit of genetics and molecular biology is carried out with it. This is, this should have been green in color. It's Chlamydomonas, which B.J. Rao worked with apart from bacteria. And so the topics or the models kept coming back and forth for TIFR. Again, in 2016, we were 14 at that time. The academic strength, if you look at postdoctoral fellow students, it was 107, of which 56% were women. Faculty was 43% women, and this is probably the best figure for institutes all over India. Typical representation in a good place is of the order of 25 to 30%, even in biology. Hosur was talking about interconnection. And I'm glad because somebody did this, I'm not sure, Vidita probably made this slide, and you can see the interconnections between the people at that time and the kind of work that people were doing. And each group interacted with the other, and there were each individual scientist interacted, and then there were common publications quite often. Subsequently, many of us have left, and new members have joined in. Devashish Das, Amitesh Anand, Nitin Singh Chauhan, and Sundaram Naganathan. And so somebody will have to make that chart all over again and look at whatever fields they are studying. So the strength of Department of Biological Sciences has been between 10 to 14, and the kind of space it has at the moment can accommodate no more than that. Achievements, very difficult to talk about achievements, you know. How do you judge achievements in fundamental science? Mainly publications, right? So I have listed the kind of journals that we have published in from molecular biology and DBS, but it is representative of all of TIFR to publish in these journals. And I will pick up a few and talk about them. But I wanted to highlight publications arising from interdisciplinary research within TIFR. It has been heavenly for interdisciplinary research. We've had close interactions with the physics and chemistry members in the canteen and everywhere. And such fallouts have been magnificent. So these are the publications that I've listed. This is also till 2016, there might have been more. So people must excuse me for such limitations. Okay, what I wanted to point out was also extramural fellowships and grants. So this is something that biologists have always sought, because money was always limiting, and it was also prestigious to get some of these grants, and they are extremely competitive. So this is again a list from 2011 to 2016. So it doesn't include many of the earlier grants also, but you can see that, the total funds for those years did reach a very considerable amount at that point, which helped in bringing in a lot of equipments and consumables. There's a huge amount of expensive consumables required for molecular biology, for any kind of biology research. So now I'll just pick up some papers. I must say that every individual was publishing very well. We are talking of 75 years. I can't think of anyone who hasn't published well during this period. At least there have been peak times when they have published very well. So these choices which I've made, and these are from MBU DBS, are my own selection. And for Obaid, I picked out this paper and this paper. So this was his bacterial work, where Vijay Sarthi actually showed that, and Obaid actually showed that DNA synthesis is not necessary when you're having bacterial conjugation. When bacteria conjugate, DNA from one bacteria go into another bacteria through special cell biology mechanisms. And at that time, there was a question as to, did it happen with new DNA synthesis, or did it not require DNA synthesis? So these two papers, there are two papers in JMB. They are seminal papers for that at that time, because it was new knowledge really. This is also a work which was very remarkable. I mean, the fact that they were studying mutations which did all kinds of funny things to chemosensory perception was common. There were many papers published by Veronica also, and so this paper has Veronica as the second author, Kavita Arora, Veronica Rodriguez, and 2-3 others, Obaid Siddiqui. So this essentially describes work in which, for the fruit fly, a salt repelling receptor gets changed to attractive receptor using certain mutation. And then they have characterized that mutation as to what that is. And that was one of the early studies of this kind, where you can change from dislike to like for your receptors. It would have long fallouts. All those people who don't like bitters, then you can change your receptors and start liking it as sweet receptors. So, but TIFR has been rather poor on, and it's not their mandate to take it to applied sciences. As Ravi was pointing out, maybe futuristically one should look at that, but taking it to any kind of industry was not TIFR's mandate. So MM Johri and Shaila Desai, they looked at this MOS differentiation and showed, using those liquid cultures, that indole acetic acid regulates the differentiation from one stage to the other. And this they showed with very beautiful biochemical work as to what exactly happens when these things differentiate. So the very nice pictures in which chloronema has certain cell property, and then the cell wall becomes oblique. How does the cell wall change from vertical to an angular one? These are all famous cell biology problems, and he has addressed some of them. Krishnan initially worked on snail, which snails went everywhere, but I think his work did not go anywhere. But so he moved to Drosophila and he studied anesthetic response. And this was again very elegant. So using homemade funnels so that you have 5 minutes, okay. You have flies which you put them on top, and those who are normal, they will just come down. Or maybe it's the other way around. You put them in the bottom and they will naturally go up. And those who are, if you put chloroform, those who are those who will get anesthetized will fall down. But mutants which are refractory to anesthesia will continue to go up. And he has characterized several of them on that line, and he has moved on to many other aspects. Krishnan was also very restless. He moved from one project to the other. Maitra and Lobo, I cannot pick one paper because there are dozens of papers, but they did outstanding work on glycolysis, and so did Kinkare on enzyme brain hexokinase. Okay, some more recent examples. I do not have time to describe them. So, you know, you can, I mean, you can see that these are all very, very good. These are just examples of excellent work, which I would have liked to have highlighted for each one of them, but I'll just go ahead. Research at NCBS. So NCBS has a faculty strength of about 40. And each group has 7 to 15 lab members, which is really the kind that one would like to have for any effective biology, experimental biology research. It has enormous strength in these traditional subjects, which is what I have described for DBS. And in addition, it has built strengths in ecology and evolution. And you can imagine that whatever I have described for DBS, NCBS has 4 times that in terms of its prominence, not prominence, in terms of quantitation, publications, and in terms of you know, its impact in some sense, because they have huge labs. And so I cannot talk about NCBS. Somebody else has to come and talk about NCBS, because you know, it's an institute by itself. So, and a fantastic one with doing fantastic work. So, but I would like to mention 3 programs, actually 2 main categories. One is the wildlife training. So this is unique. This is unique in this country. It trains people to do wildlife biology conservation, has also worked on it at the molecular level. So there are wildlife programs, and you can join some university or other centers where you can follow the animals. But here you have the opportunity of taking samples and then working with them in the NCBS biology labs. And it's a very successful and unique program. There are students clamoring for it. I think something like 12 to 15 students are selected for the master's course, and it's very competitive. The other programs, these are centers and programs of NCBS, which I have picked up. One is chemical ecology, and this is in collaboration with scientists elsewhere. Currently, it is only Balaram and Reni Borges from IISc, but it clearly plans to expand. I couldn't resist the temptation of the beautiful pictures that were on their web pages. And they look at molecules exuded by them or secreted by them and look at their chemical composition. And I would have liked to have added cone snail, which is what Krishnan was working with, but I couldn't find a beautiful picture. So, but that was another one of these. And Krishnan, I mean, this was started a little later, so it's following the tradition of Krishnan. This other program is that of ADBS. This was launched in 2016, and this is using NCBS, InSTEM, and NIMHANS. And this works with, so Shobhona Chatterjee is the major driver. Deepa Agashe was the major driver in the earlier program. Shobhona Chatterjee is the driver of this. And they essentially collect material, human material, to look at genetics and genomics and, you know, look at mental disorder. And right now, you know, it is still in the data collection stage, but it is expected that this will give enormous value. This has enormous value and will be enormously useful. And yeah, I'm almost 2 more slides, sorry. So this is the third one, which is a lipid research, Max Planck NCBS Center on Lipid Research. And you can read the thing, and I like the logo. So these are membrane molecules, you know, bipolar. So hydrophobic head followed by tails, and then the keys which can unlock such such properties. And some of the distinguished alumni. So this I've just listed over here. This was also one of the 2016 slides. So it just lists people with their accolades. I haven't talked about awards because they have been, you know, you name it, Infosys prizes and Patnagar awards and so on. They're both for DBS and for NCBS. And I just wanted to list alumni with applicable achievements. And I wanted to, of course, talk a bit about my current work. So Handa, who was a student of M.M. Johri, he went to Purdue University, and his work gave rise to genetically modified tomatoes with high pectin content. And these are in the market, and they are great favorites for ketchup production. But of course, you know, the inventor's name never doesn't come up, but you feel so satisfied. I mean, he told me that I feel so happy that my tomatoes are in the ketchup, but you know, that's what it is. And we are trying to test out nanolipid carrier mediated formulations for antimalarial delivery. And we have the work done in collaboration with ICT and TIFR using animal models shows very clearly that it can be used at about 1/5 concentration. So right now there is a lot of weightage on people because they have to take combination doses, 2 tablets 3 times a day, and they have a lot of side effects. So you can reduce it to 20%. And that such combination drugs with the use of these carriers can be delivered intravenously, which can't be done as of now. That's very important because in cerebral malaria, patients go into a coma, and then you have to put intravenous drips, and you can only use artesunate, which is soluble, so it becomes a monotherapy. WHO has mandated combination therapy, but for those patients, you cannot help it, it's a monotherapy. So we have shown that okay, here, sorry, I have 2 more. So we have shown all that, you can look at. Workshops and conferences, you know, Mahabaleshwar seminar is one I wanted to point out because our department keeps asking for money from TIFR to conduct this. It's a very successful conference. Some of the visitors at MBU, traditional to TIFR style, fantastic workshops. So you have, I think this is Maurice Fox and Frank Stahl, the Michelson and Stahl fame. He came and he was so much into Indian, this thing that he would confer. You can see Obed over here. So he got into Indian dress and gave degrees to students. And of course, Watson's visit to TIFR and to labs also. Then some of these labs, and you know, I wanted to show this. This is Maitra Udaynarayan Singh, B.N. Apte, one of the first few students of MBU, and Kinkare. The rest of the, and Kakeri, who was our engineer. The rest of the people I don't know. This is N. Gautam doing experiments on ultra centrifuge, huge ultra centrifuge, and SM Joshi doing some experiment. Frank Stahl teaching MS Shaila, who was a faculty of, who's now retired, but was a student in that workshop. So thank you very much. I don't have time to explain those colorful slides, but